All right. Hey, everyone. It's great to be here in person. Happy Juneteenth for those of you who are celebrating, and happy Father's Day for those of you who are celebrating as well. Okay, so today I'm going to talk about neural encoding models. I'm going to define what that means in a second. But before I do so, I want to do a quick PR. So I'm not sure if you've heard, but Cornell has a new campus in New York City called Cornell Tech. And that's where I'm located. I actually moved there about a year ago from our main campus, which is in upstate New York in Ithaca. Cornell Tech has a very sort of strong emphasis on real world translation and real world impact. And as such, there's a real sort of focus on healthcare applications of AI in general, but computer vision also. I also have a secondary appointment with Weill Cornell Medicine, which is also a campus of Cornell in New York City in the Upper East Side of Manhattan. And we're hiring at all levels. We're hiring grad students, so you can apply to Cornell's PhD programs. We're hiring postdocs, and we're hiring faculty members as well. So feel free to reach out if you're interested. Okay, so what is a neural encoding model? So this is the setup we're interested in. You have human subjects lying in a scanner, an MRI scanner, and we're requiring functional MRI data. This could be other types of functional imaging as well, but in this case, it's going to be functional MRI data. And they're being exposed to stimuli like visual stimuli or auditory stimuli. In this case, it could be looking at pictures. And as they do so, we're collecting data. We're collecting data that reflects the brain activity. In the case of functional MRI, that brain activity is what we call the BOLD response, or the blood oxygen level dependent signal, which is a proxy for the sort of the neural activity in a voxel or in a 3D pixel. A neural encoding model is essentially a model that takes you from the stimulus and predicts what the brain response is going to be. And you basically train it on some data collected in this way. So there are two parts to this. There's one part which we call the feature extraction, where you basically take, you apply this to the stimulus, in this case, an image, and it extracts the features from the image. And then there's the model that sort of takes those features and turns them into a brain response prediction. Okay? And the reason we want to do this is we want to gain insights about sort of how the brain works, particularly the visual system in the brain. Okay, and why do we want to do this? Well, there's basic neuroscientific reasons for understanding how the brain works, but also it can inform machine learning and computer vision as well. So that's sort of the big motivation. There's been quite a bit of work in this area. So sorry, before I go there, the opposite of this, so going from the brain response to the visual stimulus or just general stimulus or the behavior, is what we call a neural decoding model. And you can imagine that that has a lot of practical applications, like in the context of brain-computer interface. Okay? That's not something I'm going to talk about today, but that's something we work on in the group as well. Okay, so there's been quite a long sort of list of papers on this topic that goes back over a decade. I think one of the earliest papers that I really enjoyed reading was by Jack Gallant and his students at Berkeley, where they actually focused on the decoding problem more than the encoding problem, and looked at sort of fMRI data to understand or to predict what the human subject was looking at. And then there was more recently some work by Jim DiCarlo at MIT and his students, Dan Yemens in particular, who applied sort of more modern neural network style techniques to the encoding problem. And essentially what they did in this sort of line of work was to show that computer vision models like the famous AlexNet or VGG or ResNet, whatever, trained on computer vision tasks, if you take them, those pre-trained models, and you looked at the features in those pre-trained models, you can map those features linearly onto fMRI response. So, and in different parts of the brain, that mapping looked quite different, and sort of you could gain some insights about what's going on in the brain. So this was sort of the literature we were aware of and we were thinking about, but there was some important gaps in that literature that we wanted to sort of look into. And one particular gap I'm going to talk a little bit about is attention. So the one open question that we were really motivated by was, can we sort of, if you think about human visual perception, it's really modulated by attention. I'm going to give you an example in a second. And you can actually measure that attention through eye gaze data. And so the question was, can we leverage eye gaze data to improve the prediction of our neural encoding models? That was one question. And the other question was, well, assuming that we don't have eye gaze data, can we build an attention mechanism into our neural encoding models that is sort of mimicking eye gaze data, and then see if that agrees with eye gaze data in a subsequent analysis. Okay, so what do I mean by attention? Well, it's really well established, right, that human visual perception and also auditory perception is actually very heavily modulated by attention. We basically see what we attend to, and there are many famous sort of human psychological and neuroscientific sort of experiments that demonstrate this, but you can immediately see this in this simple example where, the two images, it takes a couple seconds for you to figure out what the difference is between those two images, because your attention is drawn to the salient object in the center, and you sort of miss the detail that's sort of different between the two images, which is on the upper side, that branch there. So essentially that sort of demonstrates how attention is driving our perception of a scene, okay? So this is what the data is going to look like that we're going to train on. So as I mentioned, human subjects are lying in an MRI scanner and they're watching a movie or looking at pictures. In this case, they're going to be watching a movie. And as they're watching a movie, which I'm going to show on the right, their brain is responding to that. And so we're measuring the brain activity through a functional MRI. And we can pre-process that functional MRI data and project it onto what we call the cortical surface map, and visualize that. So anything that's blue is underactivated, anything that's red is overactivated, and that sort of gives us insights into which parts of the brain is responding to the stimulus. And on the movie frame itself, you'll see a bunch of dots, and this movie was watched by a few hundred human subjects, and each dot corresponds to the eye gaze of that human subject, and the big circle will be average eye gaze data. So this is sort of what the video looks like. And as you see on the left, you'll see sort of the slow moving fMRI response, and it's slow moving because even though the underlying signal is very rapid, it's slow moving because it's modulated through what we call the hemodynamic response. The blood, the blood oxygen level dependent signal is a sluggish signal compared to the neural activity data. Okay. Does anyone recognize this movie? No? Nobody. Wow. No. Okay. Was it Aaron Brockovich, right? That was the movie? I think so. I'm hesitating right now because I'm kind of, maybe I got it wrong. I don't know. I think it's Aaron Brockovich. All right. So that's what the data looks like. Okay. So what is the model going to look like? Okay. So in this particular project that I'm highlighting, we will use a ResNet backbone as a feature extractor that's being pre-trained. So we won't touch ResNet background. It's a ResNet 50 that you input the image to, and then it spits out a pixel grid, a lower resolution pixel grid with 2048 number of channels, and that's our feature that was extracted from the image. And then what we're going to do is we're going to take these features and map it to the fMRI response, and there's a couple ways that we're going to do that. One way is we can take the eye gaze data that we've measured, and actually in this case, it's just going to be the average eye gaze data, and turn it into a Gaussian blob, okay, where we have a mean and a sort of a standard deviation. And then we're going to multiply that element-wise with the feature map, And then that's going to be, and then we're going to basically pool that over space. So it's going to be some attention weighted average pooling over space, which is going to yield 2048 dimensional feature vector that we're going to feed into another model, which is a convolutional neural network that turns that into a 3-dimensional brain response map. Okay, and so that's one approach. And in this approach, the key thing is that we're using eye gaze data explicitly. Another approach, we're not going to use the eye gaze data, so we're sort of ignore that. Instead, we're going to say, okay, let's try to learn an attention network that takes this feature map and figures out where to pay attention to in the image, and then use that to multiply with, again, the same way as we did before, with the features, and then turn them into a 2048 dimensional vector through the attention weighted pooling, and then again use the same exact response model. Okay, so we have two versions of the same model. One uses eye gaze data explicitly, and the other one does not use eye gaze data, but tries to learn what to pay attention to in the image. So this is the data. It's 158 subjects. They're watching a bunch of movies. I'm not going to go into detail. We have training, validation, blah, blah. And one important thing I want to highlight here is this is a population level model. We're learning a single model for everybody. Okay, it's not individual specific. So it's just one model for everyone. And this is just a very quick summary of some results, right? So you can quantify the accuracy of a prediction by looking at the agreement between the predicted brain response and the measured brain response. And there's different ways of looking at it, but a very common strategy is just to correlate using Peirce's correlation. And what we sort of observe at the sort of the course, high level, is that using eye gaze data gives you the best result in terms of predicting brain response. And then the learned attention mechanism also gives you a pretty good result, even though you're not explicitly using eye gaze data, it somehow figures out to exploit that attention mechanism and improve its prediction accuracy. We have other baselines here that are not doing as well. One particular baseline that I'm going to tell you is the center attention, which is basically a fixed attention in the center of the frame, assuming that everybody just fixated into the center of the frame, which is not the case, obviously, because of the way we're perceiving movies or watching movies. We can look at the accuracy at different parts of the brain. This is sort of, again, an inflated cortical surface, looking at it from the back. So this is the occipital lobe of the left hemisphere. And these are the different sort of visual areas, the higher order visual areas that are mapped up. And we can sort of quantify the quality of your prediction model in different areas. And one thing that's sort of striking to us, at least, is that in higher order visual areas that are doing more and more semantic or object-oriented processing, you're getting a bigger boost when you use the visual attention mechanism. Okay, lower order areas don't really benefit that much from visual attention. If you compare the fixation, the eye gaze tracking data to the learned attention, okay, there's a striking similarity. In my mind, this was the most important result of this particular paper, where basically the model itself learned an attention mechanism that is exactly correlated with what humans are paying attention to in their eye gaze data, even though it's not explicitly using eye gaze data, and it's only tasked with predicting the brain response directly. I guess it's unsurprising retrospectively, but it wasn't something that we expected, at least this degree of agreement. So what we are showing you here is the human fixation data. So these are the dots here, and these are the predicted attentions, right? So this is the learned attention mechanism of the model. So you can do a sort of there's a quantitative analysis of this in the paper that was published a couple of years ago in Neurics, but you can do a quantitative analysis and show that these are in very strong agreement. Okay, so just a couple of remarks before I move on. Gaze weighting helps a lot in terms of trying to predict the attention, to predict the brain response. But in the absence of gaze data, which eye tracking data is not always available to you, you can actually build explicitly an attention mechanism into your neural network that will learn what to pay attention to, which will also be correlated with eye gaze data, but B, also improve your prediction accuracy. accuracy, because that's how our brain responds. Now, the big caveat of this work that I've presented so far is that it's at the population level. As I mentioned, we have a single model for everyone. Okay, obviously human brains are different. There's a lot of individual variability that we're interested in neuroscientifically, and the question was, can we explicitly characterize that variation using these types of approaches? Okay, so this work, which formed the PhD thesis of a student of mine who I'm going to talk about at the end, has 4 parts to it. I only talked about this particular part where neural encoding visual attention. I'm going to skip this part, which appeared last year in a journal paper. I'm going to talk briefly about this part, which is individualizing these types of encoding models. How would you do that? Okay, so why would we do individualized encoding models? Well, first of all, we're really interested in subject level characterization of the brain, right? We don't, population, neuroscientifically, obviously, population level characterization is interesting, but we really want to drill down into individual level variation. The challenge is fMRI data is famously low SNR, and we really don't have the luxury of collecting a lot of data per individual. We really want to minimize that data collection requirement. Okay, so we want to be able to get away with like a 5-minute scan or a 10-minute scan, not have subjects lie in the scanner for hours and hours on end. Now, the approach we're going to take is we're going to leverage the group level data. We have lots of data from different individuals. We're going to leverage that and try to build a subject-specific model that sort of takes advantage of the group level data. Okay, so this is one way of doing it. So in this approach, we have 2 parts in our model. We have the so again, we have the features that are extracted by your feature extractor. Let's say a ResNet 50. And then you have a part of the model. The first part of the response model is what we call the coarse response model that's shared across individuals. So it takes this feature vector and turns it into a coarse fMRI response at the population level. And then we have subject level refiners. Okay, so these subject level refiners basically takes this fMRI response of the population, the predicted fMRI response for the population, and turns them into a subject specific high resolution fMRI response using transposed convolutions. Okay, and this part is unique to the subject, unique to that individual. And this first part, the coarse response model, is shared across individuals. Okay, so this will allow us to pull data across individuals, learn this efficiently, and then there's, not that many parameters left over to fine tune for each of those. Okay, so here's some sort of very high level results. We can quantify, basically in this case, we actually looked at 2 types of neuroencoding scenarios. One was when we have auditory encoding, so they're listening to something, the subjects. And we're sort of trying to predict what their brain is where their brain is going to respond. And then there's visual encoding, like where they're looking at pictures. And we see that, so the blue sort of results here, the results that are for the model that I just showed you, where there's a shared response, a population of response, and then individual refiners. The pinkish model is essentially a model that is only is trained for everyone. It's a population level model. This green model is the same architecture essentially, but you train it individually for every subject. So you train it from scratch. Okay, the neural response part, not the feature extractor. The feature extractor is frozen. And then we have this gray part, which to us represents the state-of-the-art baseline, because it takes the feature extractors, the frozen feature extracted features, and uses a linear model on top of those to predict the brain response, which is what pretty much everyone did until our work. And if you look at this linear model, it does reasonably well. It's not terrible, but you can obviously outperform it, especially for visual stimuli using CNNs. And you can see that there's sort of the best performing model is a model that can pull across individual subjects. Now, the pink model, the shared model, which is the population model, is not too bad. you might argue that why not use this? The challenge is this is not capturing anything about individual subjects. It's just one big model for everyone. And if you wanted to study intersubject variability, you won't be able to capture that because it's the same for everyone. So we were interested in looking at intersubject variation, and so we took this R model, and basically it spits out a predicted response, and then there's a subject measured response, and you can correlate them between subjects. So you can get this matrix, which is number of subjects by number of subjects, and each entry in this matrix is the correlation between the predicted response for that subject and the measured response for the other subject. Okay? And the diagonals are for the same subject. And you want this to be a diagonally dominant matrix. Okay, if your model's capturing intersubject variability, it needs to be diagonally dominant, and obviously we see that here. What else could we use this kind of model for? Well, now we have a model that can take any arbitrary stimulus and then predict the fMRI response for an individual subject, right? So we can treat this as an in silico brain, right? And then you can essentially replicate any fMRI experiment you want on your computer. So you can show a bunch of faces and non-face images, and you can compute these contrasts. You can show its images of scenes and images of humans and images of cats and dogs, and basically do your fMRI mapping experiment in silico using just this synthetic brain. And that's what we did. And what we sort of found is that you get a lot of, so you can replicate what people have sort of discovered over many, many decades of my research, right? So fMRI has been around for, I guess, two plus decades now. And you can basically, and people, the way they use fMRI is you typically hypothesize that there must be a brain region responsible for detecting faces, right? And so you show a bunch of people face images and non-face images, and then you contrast the two to figure out what parts of your brain is selectively responding to faces. So it's a very hypothesis-driven approach, and you can imagine that that approach is limited to what your hypothesis is, and every time you have a hypothesis, you have to collect new data, okay? But the beauty of having an in silico synthetic brain is that you can basically test unlimited number of hypotheses without really having to go and collect data first in your computer, and then maybe to verify your results, you can go and collect data on some real individual subjects. Anyway, bottom line is we could replicate the phase contrast, the famous phase contrast in fused form area. We can also replicate the scene contrast. There's a bunch of other replications that exist in our paper that you can go and look at. Okay, so this to me is a nice step towards developing encoding models that have a shared piece that is at population level, but also can account for individual variability. And one very strong utility of this type of approach is that it gives us a way to synthesize fMRI data and conduct these sort of in silico experiments that would be otherwise hard to do. Okay, so one final thing that I want to highlight or talk a little bit about is more recently we were really interested in can we do other things with our neural encoding models to gain further insights into how brains function. Okay, and I talked about attention. I talked about individualizing these models and using them as synthetic experimenters. But we could, in this particular work, we were interested in can we design this particular piece of the neural encoding model, the piece that predicts the response of the brain, in a way that could give us insights about, if you look at especially visual neuroscience research, a lot of research is about characterizing what and where the brain is a particular brain region is responding to in visual scene. Okay, so there's this what and where dichotomy, if you will, which sort of maps nicely onto the two visual streams that people have characterized. Okay, and so the way we sort of thought about approaching that in this sort of what and where framework is that so instead of predicting some average response for a given region of interest, we're going to do voxel level prediction now. And for every voxel, which is a pretty large region in the brain, might have like tens of thousands of neurons in it, what we will have is we will have a visual attention mask. So it's going to, every voxel will have its own attention, and it will also have its own sort of spatial pooling layer. So we have the same feature extractor backbone, but the way we're going to combine these features into a brain response for every voxel is it's going to be a spatial pooling that will correspond to that voxel's attention mask times, and this is an outer product, times a feature attention mask, right? So over channels. Okay, so that's basically how we decompose the neural response model, and every voxel will have its own, and we also learn those by sort of data-driven training. So for this particular experiment, we use what's called the natural scenes data set, or NST, which is a pretty rich data set that just came out, I guess less than a year ago now. That involves 8 subjects looking at natural scenes from the COCO image data set for about 40 hours each, or over 40 hours each. Okay, so it was a little, I guess, a little bit of a torture for these subjects to lie in the scanner for that long. It was spread out over many months, but it's a very rich data set. It's narrow in its scope in terms of number of subjects, but it's for each subject, there's a lot of deep data that's being collected. Okay. Okay, so we can look at individual subjects in this data set, and the model that I talked about, this model here, when you look at how well it fits these subjects and you project it onto the cortical surface, these are flat maps now because we want to visualize them, sort of, these nooks and crannies of the cortical surface, the visual cortex. The prediction accuracy is really nice. So we were very happy about how well we predicted in different parts of the cortex, and this is sort of partly a function of the amount of data we have access to in this data set. Okay. Now, one thing we were super interested in looking at for this is one of the fundamental questions about characterizing the early visual system in the brain is there's this retinotopic mapping between the visual scene that you're looking at and the early visual system in your brain, particularly V1 through V4. Where basically every little neuron in the visual, early visual system corresponds to a particular location in your visual scene that you're attending to. And one way to characterize that is for every neuron or every voxel you can think, there's an eccentricity and a polar angle. Okay, in the visual scene that you're looking at. And the way you map it is through this so-called retinotopic mapping experiment, where you have a checkerboard disk that is expanding and a checkerboard wedge that is rotating. And you can sort of take these two artificial stimuli, present them to human subjects, for let's say 10, 20 minutes. And then using those data, you can actually map every particular voxel's retinotopic sort of eccentricity and polar angle. Okay, and this is a classic fMRI study for characterizing early visual cortex, the sort of V1 through V4. The question we were asking is whether we can use our neural encoding model to actually get to these retinotopic maps without having to show artificial stimuli. Okay, so what we did was we actually took, if you remember, in this model, we have an attention mask for every voxel. So we can take this attention mask that's been learned by training on the data and try to fit it and figure out what the eccentricity and the polar angle will be for that particular voxel. And so we have an estimated eccentricity and a polar angle. And we can, and in these subjects, we also have retinotopic data. So we actually have measured eccentricity and polar angle. And we can look at it as in a scatter plot like this. And V1 through V2 through V4, we observed that there's a strong agreement between what's being measured in the retinotopic experiment and what we estimated using just natural scenes data. Okay. Same goes for the polar angle. And one big question we had was, well, the natural scenes data has a lot of data. As I said, it's over 40 hours of scanning. And, it's interesting in theory that we can fit a neural encoding model onto this 40-hour long data, and we can then interpret this neural encoding model in a way that it estimated the retinotopic mapping of the early visual cortex. But what practical utility does this have? A written topic mapping experiment's only like 10 minutes. So I can get that map, those maps in 10 minutes of just showing those artificial stimuli, and I'm done. If that's what I'm after, why would I go through this exercise? Well, one sort of, so we that was a curious thought experiment. So what we did was we actually changed the number of images that we fit on. So instead of using the entire data set, we went down to about 100 images to train our model on. And then we looked at the correlation or the agreement between the estimated and the measured written topic parameters. And we noticed that, with about 500, let's say, you get really good agreement between the two. And 500 is going to correspond to like a 20-minute scan. Okay, so it could be practical. Okay. Another thing we were interested in doing in this case was, remember I said one big question is what and where in neuroscience. Here we were sort of looking at what, sorry, this was where, I'm sorry. So in the early visual cortex, each voxel, where is it ascending to in your visual scene? And we were sort of, the estimates we got, we were comparing them to the written topic maps that you measured. Another question, as I said, we can ask is what? So, and this is particularly interesting in later stages of your visual cortex, especially in VO. And one sort of, I'm going to show you one result here. There's many results that are in our paper that's under review right now. But one way to look at this was you have the feature attention map, right? So the neural decoding model, or encoding model, I'm sorry. You can take this model that you've trained for an individual subject, and you can input any images that you want. In this case, we took the things data set of 27,000 images of different things, of different objects. Pushed them through our neural encoding model and looked at that attention map for each one of these images for each voxel. And then we did a PCA on that. So it turns out that with two principal components, you can explain about 70% of the variation on average for every voxel. And then for VO cortex, on average, we basically looked at the first principal component, and we looked at the images that activated the first principal component most positively and most negatively. So the top row is the most positive images, and then the bottom row is the most negative images. And one striking interpretation of these results is this first principal component corresponds to inanimate versus animate, that dimension in VO response. And this is very well studied and well characterized. Okay. The second principal component turned out to be rectilinear versus round features. So it corresponded to a shape dimension, which has been speculated. And there's a couple of papers out there that talk about VO being responsible for shape interpretation, but it's really not nailed down in neuroscience. There's a lot of debate going around this. So this to me is a very striking example of basically providing us with a novel way of looking at data that might allow us to corroborate a certain hypothesis. In this case, VO being responsible for shape analysis. Okay, so I'm going to stop here. But before I do so, I hope you're convinced that neural encoding models could be interesting and give us a novel tool to do some basic neuroscience. A lot of this work obviously was done by Meenakshi Khosla, who was an amazing grad student in my lab who graduated last year and is now at MIT with Nancy Kenwisher doing a postdoc. Gia also did a lot of the work with Meenakshi, and Amy is a fantastic collaborator of mine who basically does a lot of really good fMRI research. I would also like to note that my group does other stuff as well in vision. Today I decided I want to talk about neuroscience because I thought it might be interesting to this crowd, but we do a lot of registration segmentation kind of work as well. So anyway, if you're interested in any of this, please feel free to reach out. Thank you.